Thank you to the organizers for inviting me to speak today. Today I'll be discussing the vaginal microbiome and HIV acquisition and its interactions with hormonal contraceptives. I have no disclosures. So it's always nice to start a talk with a nice picture. What we're looking at is a picture of the gut of a mouse and the microbiota sitting on top in purple and red, sitting on top of a thick mucosal layer in green, atop of epithelial cells in blue. And when you look at this picture, you can appreciate a couple of things. One, that we're vastly outnumbered and that bacteria are living in all surfaces of our body. And also that the microbiome is in very close contact with mucosal surfaces, and these interactions are very important. The microbiome is very important for our health, as we all know. And one concept that has been linked to many chronic diseases is the concept of microbial dysbiosis. And microbial dysbiosis can be characterized in many different ways, including changes in metabolic functions, distribution of its members, or its diversity. And microbial dysbiosis has been linked to increased risk of chronic diseases such as cancer, diabetes, obesity, as well as cervical cancer, preterm birth, and increased risk of sexually transmitted infections. And microbial dysbiosis in both of these compartments, the gut as well as the vagina, have been linked to increased HIV pathogenesis and transmission risk. Today, we'll be talking about HIV transmission, and a very important microbiome for HIV acquisition in women is that of the vaginal microbiome. And the vaginal microbiome can be divided into two very broad groups. Those that are dominated by Lactobacillus species, including Lactobacillus crispatus, Lactobacillus inners, Jensenii, and others, which can vary considerably in their inflammatory properties as well as their ability to produce lactic acid. And then women that do not have lactobacillus instead have anaerobic bacteria, including Gardnerella vaginalis, Atopobium, Prevotella, Mobiluncus, and other facultative species. And this is commonly associated, this is what's known as microbial dysbiosis or bacterial vaginosis. Now, in order for HIV infection to occur, it must traverse the many physiological and biological barriers of the female genital tract, including a squamous epithelial barrier, a layer of protective mucus, as well as resident immune cells. However, a major risk factor for increased HIV acquisition in women is genital inflammation. And this can be broadly characterized as increased pro-inflammatory cytokines, increased number of activated T cells, or the number of T cells, which are targets for HIV, and epithelial barrier disruption. Now, the vaginal microbiome gives many functions that are important for mucosal homeostasis. Lactobacillus species can produce lactic acid, which help to keep the vaginal pH quite low and acidic. It also provides some other antimicrobial functions, and lactic acid itself can be anti-inflammatory. Whereas in the absence of lactobacillus and the presence of bacterial vaginosis, this is associated with many changes in the vaginal microenvironment, including increased vaginal pH, pro-inflammatory cytokines, epithelial disruption, short-chain fatty acid production, as well as the production of enzymes by the bacteria, which can degrade protective features, including mucins and mucosal IgA. So it's not surprising to know that bacterial vaginosis has been associated with increased HIV acquisition risk in women for a long time. With the advent of better molecular tools, we've gained a better appreciation of the relationships between the vaginal microbiome and HIV acquisition. And this is a study showing data from the FRESH cohort in South Africa, looking at the microbial diversity in young women. On the top right, we can see the plots. Some women had Lactobacillus crispatus in yellow. Many had Lactobacillus inners in green. Some women had Lactobacillus or had Gardnerella vaginalis in blue, and those in red were highly polymicrobial with no predominant taxa. An important observation here was that women with polymicrobial microbiomes were at fourfold increased risk of acquiring HIV than women with L. crispatus. And this was associated with increased levels of pro-inflammatory cytokines, as well as increased activated T cells in the vagina, and this is shown on the bottom right. Follow-up studies have also shown a dose-dependent relationship between anaerobic bacteria and increased HIV acquisition, including Gamella, Dialister, Parvomonas, and others, which independently associated with the 2 to 3-fold increased acquisition risk, whereas Lactobacillus crispatus and to a lesser extent Lactobacillus inners is associated with relative protection from HIV. Our group, in collaboration with CAPRISA and University of Washington, performed a sub-study of women that were enrolled in the CAPRISA 004 trial, which tested a vaginal microbicide for HIV protection. And the major finding was that women with vaginal lactobacillus had the drug work 3 times better than women that did not have vaginal lactobacillus. And this was associated with drug metabolism by anaerobic bacteria, as well as increased genital inflammation. However, follow-up studies showed that from the Partners PrEP trial from Renee Heffron and Jared Baton showed that there was no relationship between bacterial vaginosis and the efficacy of oral PrEP, suggesting that the modality and delivery mode of pre-exposure prophylaxis is important for whether these interactions may occur. There are over 800,000 new HIV infections in women around the world, and many of these are in sub-Saharan Africa. And the prevalence of bacterial vaginosis in many countries in Africa is quite high, and this is shown on the pie charts on the right. In purple shows women that have bacterial vaginosis, and in orange and yellow are women that have vaginal lactobacillus, showing that BV is quite high. Therefore, the promotion of lactobacillus could have a significant impact on HIV acquisition rates for women, as well as other reproductive health outcomes. Other factors also affect HIV acquisition risk, and this is a meta-analysis that shows a relationship between injectable hormonal contraceptives such as dipomidroxyprogesterone acetate and relative risk of HIV acquisition compared to other contraceptives. In these various meta-analysis, an observation was that DMPA is associated with 1.3 to 1.5-fold increased risk of acquiring HIV compared to oral contraceptives or no contraceptives. However, these studies were post-hoc analysis from various cohorts, which were confounded by other variables which were not controlled for in the original study design, as they were not designed to study this specific question, which led to the ECHO trial, which randomized women to 3 different contraceptives, including DMPA, copper IUD, and an LNG implant, which found that there was no difference in HIV risk of the different contraceptive methods between the different contraceptive method groups. However, it should be noted that this study trial design was designed to detect a 50% or greater increase in relative risk of acquiring HIV. So with these observations, there has been an abundance of papers that have looked at the effects of DMPA on the vaginal mucosal microenvironment. And this is a review that has been submitted by my student, Hosanna Ayeli. And she went over papers over the last 20 years looking at the relationship between DMPA and the vaginal microenvironment. And one of the questions that came up is, how does DMPA associate with changes in the vaginal microbiome? And while some studies showed a decrease in the relative levels of G. vaginalis or Lactobacillus inners or some of their functions, other studies saw an increase in G. vaginalis or Lactobacillus inners, whereas a majority did not find any significant changes in the microbiome, suggesting that these relationships are complex and there are no clear associations. However, studies have found that, many studies have found that DMPA associates with increased cervical vaginal mucosal immune cells, including CD4 positive T cells that are activated, as denoted by these various publications. Also, DMPA has been associated with changes to molecular pathways in the vaginal epithelium. And many papers have shown that this associates with changes in pathways related to epithelial differentiation and tight junction formation. And a picture is worth 1,000 words, and this is beautiful work done by Dr. Ann Karias at Northwestern University, where she studied the vaginal epithelium in pigtail macaques over the menstrual cycle, as well as when the pigtail macaques were on DMPA. And as you can see in this image, with DMPA, there is a loss of the thickness of the vaginal epithelium, loss of the stratum corneum, which is more pronounced than that of the luteal phase when progesterone levels are high. And this associated with increased levels of HIV target cells within the tissues of the vagina. So with all of these observations, we thought that the interactions between the vaginal microbiome and hormonal contraceptives may be important for HIV acquisition risk. And a recent study by Haddad et al. Indeed found an association between BV and HIV acquisition, in which women with BV who were on DMPA were at increased risk of acquiring HIV compared to women with BV and were not on DMPA. So we have been partnered with HIV prevention trials and leadership in the HIV prevention field to study vaginal mucosal biology and how this relates to HIV transmission. And this includes Connie Seelam, Jared Baton, Nellie Mugo, and Jay Lingappa from the Partners PrEP trial, Sharon Hillier, Jeannie Marazzo, and Mike Cherenje from the VOICE trial, and Salim Karim and Koresha Karim and Lyle McKinnon from CAPRISA. And with their generous support, we have been generating data to understand these relationships. So there's different ways that we can measure the microbiome, including looking at using target analysis to look at genes, which can give us compositional information of the microbiome. We can also do shotgun sequencing to look at the metagenome, which gives us functional potential of the microbiome. By looking at transcripts, messenger RNA, by metatranscriptomics, we can understand the functional potential. And by metaproteomics and metabolomics, we can look at translated protein products as well as the secreted metabolites, which gives us better information on the functional potential of the microbiome as these later components can interact with the host. And our laboratory uses these later techniques to look at the vaginal microbiome. So we have profiled over 1,600 women by metaproteomics techniques, and this is a diversity plot, and each dot represents a specific woman, and you can see that the distance between the dot is how different compositionally they are from one another, and including those who had L. crispatus, Jensenii, or others in the top there. On the bottom right in brown are women who have Lactobacillus inners, and the bottom left are women who have G. vaginalis, and in those in red are women who have polymicrobial microbiomes with no dominant taxa. So we did a case control analysis within the CAPRISA 004 trial, as this microbiome data had been published previously. On the right, women who are using DMPA compared to women who are using NET-N or oral contraceptives, there was no difference in the relative risk of acquiring HIV. When we stratified by microbiome type, in non-lactobacillus dominant women, we did not find any difference between HIV acquisition rates and contraceptive usage. However, in lactobacillus dominant women on the left, we saw a threefold increased risk of acquiring HIV in women on DMPA compared to NET-N or combined oral contraceptives. Using baseline rather than visit data did not change these observations. And it should be noted that within the CAPRISA 004 trial, the distribution of contraceptive use is shown on the left there, where the majority were on DMPA, while others were on NET-N or oral contraceptives, while a small minority were on non-hormonal contraceptives. And this is the plot of the vaginal microbiota of each participant in CAPRISA 004 on the top, including those women who have L. crispatus on the left, Lactobacillus inners made a large proportion, those with Gardnerella vaginalis, or those who are polymicrobial with no dominant taxa. And there was no association between contraceptive usage and the proportion of lactobacillus dominance or non-dominance. In the PCA plot in B, we saw a complete overlap between different contraceptive types and microbial composition between DMPA, NET-N, and oral contraceptives. We did, however, observe a slight decrease in the diversity index of women using DMPA compared to oral contraceptives, and to a lesser extent NET-N, as well as a slight reduction in the amount of Gardnerella vaginalis or megasphaera in women using DMPA. However, adjustments for these did not change the primary observation with HIV infection. we hypothesize that there may be changes in the vaginal microbial functional capacity of Lactobacillus species in Lactobacillus dominant women that may contribute to this observation. So by profiling the functional pathways at the proteome level, we saw no changes in the distribution of functional pathways involved in metabolism or other functions in women using DMPA, NET, and oral contraceptives or non-hormonal contraceptives. So we then measured MPA levels in serum by targeted mass spectrometry, and this work was carried out by Rafilwe Molothagi from CAPRISA, as well as Lyle McKinnon and Al Leslie at the African Health Research Institute. And by comparing MPA levels in Lactobacillus dominant women, we saw a relationship between MPA levels and mucosal proteome changes, as shown on the left volcano plot. Whereas we did not see changes in the mucosal proteome when stratified by high MPA versus low MPA on the right. These mucosal proteome changes were associated with increased pro-inflammatory pathways as shown in B in Lactobacillus dominant women, including ERK5 signaling, chemotaxis, NRF2 oxidative pathways, as well as glycolysis and gluconeogenesis, suggesting that inflammation was different between Lactobacillus dominant women by MPA levels. This plot shows a clear relationship between MPA levels and separating Lactobacillus dominant women by MPA levels. So in brown, in the brown plot in B, you can see, and in yellow are Lactobacillus dominant women, and they were separated by MPA levels. Whereas in the purple plots, there is no separation in non-Lactobacillus dominant women. Now, the protein biomarkers in C are the proteins which comprise the majority of the variance between the proteome of high MPA versus low MPA. And these biomarkers are involved in metabolism and inflammation pathways. And right beside it, many of these biomarkers are independently associated with increased HIV acquisition risk, as shown in red, including PYGL, PGLS, and ALDOC. In D, you can see a dose-dependent relationship between these biomarkers and MPA levels in serum, which increase, whereas in purple, these biomarkers were already at high levels in non-Lactobacillus dominant women and were not modified by DMPA or associated with MPA levels. We then compared inflammatory pathways in the mucosal proteome as stratified by different microbiome groups, as shown by the activation scores. And each one of these dots represents different non-Lactobacillus dominant microbiome types, including polymicrobial microbiota in red. Purple are those dominated by G. vaginalis, and those in brown are those with Lactobacillus inners in comparison to those dominated by Lactobacillus crispatus. And you can see, as denoted by the squares, many of these pathways overlapped with MPA levels, including LXR, RXR activation, acute phase response signaling, and NRF2 oxidative stress response, suggesting that inflammation pathways may be in common between those women that have microbial dysbiosis, or they may converge at some point. And this is work recently presented by Renee Heffron and Heather Jaspin, who are leading a mucosal substudy of women in the ECHO trial, looking at what happens in mucosal inflammation profiles of women 1 month post-initiation of different contraceptives. And this was a specific observation that was presented last year at IAS, where women using DMPA had higher levels of Th17-like cells in the vaginal mucosa compared to women using LNG implant or copper IUD. It is unknown whether these inflammation profiles are linked with increased acquisition risk or are related to the microbiome, and these studies are ongoing. So in summary, vaginal microbiome alterations underlie increased risk for HIV acquisition in women, and both the vaginal microbiome and DMPA associated with mucosal alterations related to inflammation, epithelial barrier disruption, and recruitment of HIV target cells. So we believe the vaginal microbiome context may be important for DMPA-associated DMPA genital inflammation and the risk of HIV acquisition. However, it must be noted that this study, CAPRISA 004, has many limitations, including that this is a post-hoc analysis. The study groups were not balanced. It was not randomized for different contraceptives, and there are differences in risk behaviors that may not have been captured in the primary study. So nevertheless, we believe that further study to understand these microbiome host interactions with hormonal contraceptives are important for women's health. With that, I would like to thank my lab, especially Laura Romas and Michelle Perner, for performing much of this analysis and this papers presented today, as well as Hosanna Ayeli, as well as our important collaborators, Salim Karim, Karisha Karim, and Lyle McKinnon and Rafilway, as well as Al Leslie, and our other collaborators which contributed to the study, as well as the participants in the CAPRISA 4 trial and our other study cohorts. Thank you very much.